great pleasure to welcome you to this evening's event. It is, we believe, the inaugural event of the President of Ireland's Ethics Initiative, which aims to inspire discussion and understanding on some of the major and most significant ethical issues facing our society today. This evening's event is held, it's a collaboration really, maybe the first, between Trinity's Long Room Hub, which is our Arts and Humanities Research Institute, and the Science Gallery. Both organisations are really focused on not only high-level research, but also a great deal of public engagement of issues of major national and international concern. And as you know, the first event in the President's Ethics Initiative is this event here this evening to explore some of the issues associated with synthetic biology, which is, as we know, an emerging scientific field that could ultimately, I believe, I'm told, permit the design of living organisms. Maybe that already is in play and happening. It's also the closing event for the Grow Your Own exhibition here at the Science Gallery, which has been an extraordinary success and really has been a very dynamic and engaging exhibition. And I believe the curator is also here this evening, so we welcome her. I'd like to introduce those who are going to be leading the discussion this evening. Firstly, Professor Aoife McLysaght, who is the professor in genetics here at Trinity, who is going to host the debate. We're very honoured to be joined also by Dr. Drew Endy in the middle there, who's an assistant professor of bioengineering at Stanford, and who is already a world-leading scientist in the field of synthetic biology. And also Dr. Hugh Whittall, who's the director of London's Nuffield Council on Bioethics, again a leading expert in ethical issues associated with biology and medicine. And the Nuffield Council is an independent body that examines and reports on ethical issues in this field. Just want to say a few words about the President's Ethics Initiative. As I said, it was announced by President Michael D. Higgins to drive a conversation, a public conversation really, about matters of ethical concern for the society. He's asked the 7 universities and the Royal Irish Academy to take the lead in this, and over the coming weeks it's going to be announced formally. Actually, we were so far ahead that we are having the first event before the initiative is formally launched. He's also planning to have a series of consultations right throughout the next 6 months, and Trinity is going to be leading on a number of those, and then a closing conference in Áras an Uachtaráin in the autumn. We're certainly going to be leading out on a host of conversations. We're going to be engaging the college's philosophers, theologians, sociologists, scientists in conversations on cyber ethics, the ethics of intercultural engagement, ethical leadership in a time of change, and a host of other ethical issues over the coming months. And the first of those, or rather the next of those, is happening on February the 13th, I believe, which is about ethical leadership in a time of change. Again, I think should be a very lively debate. I also want to really announce something that we're very pleased about. The college is instituting a new Edmund Burke lecture that we're going to have annually, and this is going to the ethics initiative of the President is going to be the context in which we're going to have the inaugural lecture of the Edmund Burke lecture, and that will happen in the spring. And we have secured the renowned philosopher and crossbench member of the UK House of Lords, Honora O'Neill, who's going to be talking about balancing rights and responsibilities. So we're very excited about that as part of the ethics initiative as well. Before I hand over to the host for this evening, I just thought, from my own perspective, I think it is really critical that we are opening this discussion about synthetic biology, and this is really the occasion where we have an opportunity to reflect on some of the ethical issues associated with scientific research. As we know, research is an inherently dynamic activity, and its successes are really critical to the economic and social and political well-being of societies. But even as major innovations continue to be announced, especially in the science and technology fields, many fundamental ethical issues continue to be debated and need to be debated, I think. A lot of the debates historically have revolved around issues like the nature and purpose of scientific research, including often a specific concern about the balancing of benefits and harm. There are a host of questions about the treatment of research subjects, and in the field of synthetic biology, I suppose the definition of a research subject is one that is ever expanding. There are a whole set of questions about the conduct of research, including the ownership and use of research outcomes, and ensuring that there are those who are participants in the research have some interest in the benefits of that research. We're also, I suppose, aware that there are a whole host of issues about what you might call social justice issues and the distribution of the benefits of research. And I suppose in an increasingly globalized world, there is a major issue, I think, for society about whether and how there can be responsibility and accountability in this very diffuse environment. And I suppose, as we know, scientific research impacts on some of the most fundamental aspects of human biological life, including the prospects for future generations. So we have, I think, as a society, a very strong investment in the responsible scientific research and the responsible use of the outcomes, and a concern about ensuring that we have the systems in place and the discussion in place where we can have the concern about the distribution of the benefits of the research. So these issues, I think, are complex, they're wide-ranging, and they're ongoing. And the challenge for places like this, I suppose, is to be a space where we can have discussion about supporting innovation and research, supporting scientific progress within the context of debates about social accountability and equity. So I'm absolutely certain that we're going to have a very interesting and dynamic discussion this evening. I hope that you enjoyed the discussion that we have, and that you will also be interested in coming along to the other discussions and debates under the umbrella of the President of Ireland's Ethics Initiative. So it's my pleasure now to hand over to Professor Aoife McLysaght. Thank you.
Thank you, Linda. So Linda picked up on a few points there. So there's discussion and understanding, which we are hoping we'll have a lot of this evening. So we are going to open the discussion up to you, the audience, several times this evening. So if you've got questions, if questions come into your mind while we're talking, please keep those in your mind. We're going to have lots of opportunity for you to ask questions. So we're looking forward to that. And it's part of this whole philosophy of having a public conversation. So this is the first event for having a public conversation on ethics as part of this President's Ethics Initiative. So I will just introduce Drew Endy. We're very, very privileged to have Drew with us here today. It's his first time in Trinity College, and he is really a leading person in synthetic biology globally. Esquire magazine named him as one of the 75 most influential people in the world, which Esquire is, of course, the definitive resource for such things, so we'll take that. And he's really been one of the first people to get really actively involved in synthetic biology. He used to be in MIT, he's in Stanford now, and in MIT he set up the Synthetic Biology Lab and this idea of a standard set of biological parts. He's got a proper term for it, but it's the idea of biology as Lego pieces that you can combine and build together into entirely new things. And so Drew is going to talk to us now, and then after that we're going to have a discussion and open the discussion up to you, the audience. So please bring all your ideas to us as well. So it's just a great pleasure then to welcome Drew Endy to Science Gallery. Thank you, Drew.
So, hello. It's supposed to go for about 20 minutes, and I better get started. It's really a pleasure to be here. You'll see this. I dearly hope I'm wanting to take away from tonight better ideas about what should be made, if anything. And again, hopefully that'll become apparent. I thought rather than give a lecture about very technical tools and whatnot, I'd leave you with vignettes and anecdotes that create memories that illustrate what's happening in synthetic biology and then return to what we might do with it. And the three vignettes I hope to get across quickly enough are shown here: aliens, computers, and beauty. So aliens. One of the questions in biology is, how come there's biology? Where's it come from? How come there's life on Earth? And there's many different models for that. One of the models is that it came here from somewhere else. So the movie Prometheus from last 2 years ago is an example of this, where aliens, in this case called engineers, show up and deposit life on Earth. Now, is this something that only comes from Hollywood, or is this something we can approach scientifically, and what does it have to do with synthetic biology? Well, very quickly, let me refer you to this paper from the 1970s by Francis Crick and Leslie Orgel on the topic of directed panspermia. And this is the idea that life, in fact, came from Earth. It was sent here by another civilization. A very interesting paper, because basically what it starts to do is look at the time scale of the universe and when life arose on Earth of order 4 billion years ago, and when Earth-like planets existed elsewhere in the universe, many, many billions of years before that. And they basically note that there's so much time between those 2 points that you could send things around and you wouldn't have to go very fast, right? You could send a spaceship at 60,000 miles an hour, but if you let it go for 1 million years, you can cover quite a lot of distance. Now, it's hard to imagine as humans being passengers on that spaceship, and they thought the same, so their proposed spaceship would carry microbes, microorganisms that could be shielded in little packages, and off it goes. All right, now, is this a crazy idea or a good scientific idea? A couple years later, right as we started sequencing genomes, the natural organisms in the environment, another paper came out. It asks, is bacteriophage Phi-X174 a message from extraterrestrial intelligence? The research paper's on the top, and the New York Times article about this is underneath. Well, what's Phi-X174? Phi-X174 is a virus that eats bacteria. And this is a schematic of what the particle looks like. It's basically a ball of proteins containing some DNA. What's very, very interesting about these bacteriophage, these viruses of bacteria, is there's a lot of them on our planet. It's estimated by the ecologists that there's 10 to the 31, 10 followed by 31 zeros, bacteriophage particles on Earth. Million per milliliter in seawater where there's a lot of microbes. That's relevant if you wanted to send messages, because you'd want your message to be found. If you had a microscopic message, you couldn't have a microscopic library. You'd need your message to be everywhere. So if we look at the sequence of the Phi-X genome, this is what it looked like originally. It had 11 genes, and it's in this circle, and each box represents a different gene. Gene A, from 9 p.m. to midnight, is a gene associated with replicating the DNA of the virus. But what's very strange about Phi-X is inside gene A, you see a different gene, another box, a box within a box. Gene B has nothing to do with replicating DNA. It's a scaffold for assembling that weird particle. And this was very surprising when it was first observed, because the biologists were saying one gene, one enzyme, one gene, one protein. How could you possibly get this nested genetic architecture? How could you evolve it? Well, maybe it wasn't evolved. Maybe it was designed. Maybe this is where the aliens put their message. Is that a crazy idea? It's a hypothesis. Well, when you look at the DNA sequence for gene B, you can count the number of letters, and it turns out there are 121 codons that make up gene B. 121 is an interesting number. It's a square of a prime number, 11 times 11. Oh. What's the chance that the first genome human beings sequence on Earth, the DNA genome that we sequence, has genes within genes, and the nested genes are squares of prime numbers? Maybe that really is the alien message. So what these researchers did was they invented, they were the alien codex for coding information, and they made 11 by 11 matrices and tried to find the message. And this is figure 3 from their research paper. Does anybody see the alien? They concluded about the same. They said, we do not find evidence of aliens in this natural genome on Earth. Now it raises the question, and here we turn to synthetic biology, could you code other things? And the answer to this is obviously yes. Right, so here, for example, is a homework assignment from a first-year undergraduate at Stanford, where she's given DNA coding capacity and gets to code whatever information she wants. In this case, she likes to encode the Stanford logo, little s with a tree. Okay? And you can assign different number schemes to different colors or whatnot, and then you can turn that into a bit code, a binary digit code, and then you can assign bases of DNA to different bit combinations, and now you have a DNA sequence that encodes that pattern. And then you synthesize that DNA from scratch, and now you have that information in a physical molecule of DNA. What does it do biologically? We don't know. It's storing information. If you're a Harvard professor, you can write a book and then chew up your book into bits and then synthesize the DNA encoding your entire book. Right? And similar work was done here in the UK with the works of Shakespeare and whatnot in the last year. So DNA as a storage tape, and as a storage tape for storing whatever information we want, where we can synthesize or print the DNA from scratch, is really, really powerful. Now let me return it back to science quickly. Here's the map of the genome discovered by the geneticists circa 77. This year, when we relook at this genome with the power of computer analysis, we see a very different map. It's much more complicated. The boxes on the outside that are in black are the 11 charismatic genes that were found by genetics. All the other colored boxes are the ones we're identifying by computer models. We don't know if they're real or not. They all have valid start-stop signals, they look good, but nobody's ever found expressed molecules associated with them. So what we've done using synthetic biology is we redesigned the genome to erase on the phi-X genome as many of these hidden genes as we can, and we make a new genome called CleanX, where we've removed this genetic stuff, we just don't know what it is. And then we build that DNA and we put that DNA back into cells and we see what it does. So this is what the DNA does in cells normally. It grows holes in bacteria. If we unpack everything, it still works. It grows holes in bacteria. But if we erase all the hidden genes, it barely grows at all, implying that there's more to be discovered there. And by using the tool of building, of synthesis, we can very directly and quickly perturb these natural systems and figure out what we're missing. And now we have the puzzle of going back and trying to find the actual explanation. So let me zoom out. That's the first quick vignette. For your consideration, we are the aliens. We can write into DNA. We can write into DNA any message we can imagine. We're not very good at composing meaning into DNA. We're very good at printing DNA, but we can get a lot better with the grammars. And that obviously sets up the question, what do you want to say? What do we want to say? Okay, computers. Now a lot of times we think about computers, and I'll sort of test you on this, and we're sort of stuck in the present. So I want to ask you to look at this photograph and all the natural materials in this photograph. What would you build a computer from? Sand. Because it's got silica. That's right. And we do that. We got really good. Okay, so 2 points. If you build a computer from silica, from sand, you don't take the sand right off the beach and pour it on a table and expect it to be a working, reliable computer. You have to really change and rework the materials, refine it, and improve its characteristics and your ability to code stuff into it. It's not just a direct take from nature and deploy. There's a big chain of changing directed by human activity. The second thing, so the reason I emphasize that is imagine that process of transformation applied to living matter. Okay? That we don't just take biology as it exists and do something with it, but we actually rework the materials significantly. We have barely begun to do that, but that's at the essence of synthetic biology. The second from this slide is, I want you to imagine building a computer not from the silica, but from the biology. And in particular, since we were just talking about bacteriophage, these invisible viruses that are in the waters of the ocean and whatnot, you can't see them on this photograph, but I want to give you an example of building a computer from bacteriophage. And we're not going to be able to do this in the same way that the bacteriophage work in nature. We're going to actually have to take bits and pieces of the phage and rework them and assemble something new. Okay. Now, why do we think this might be possible? I want to start with nature, and then I'll move into engineering. What's shown here is a movie of E. coli cells growing and dividing. And some of the cells are infected with a bacteriophage. This is a movie, a time-lapse movie under a microscope. The cells that are infected with the virus turn green. So in this movie, there are 2 cells that are infected. One in the top right, the cell turns green and pops. And that means it was infected by a virus, and the virus blew up the cell and released more virus particles into the environment. But in the bottom middle, there's another cell that's infected, it turns green, and it doesn't pop, it goes dormant. It's the same virus. But in one cell, it realizes a different outcome compared to the other. This implies that there's a switch, a binary switch inside this natural system. Switches are parts of computers. If we could extract the bits and pieces of the phage associated with making decisions like this and redeploy them, maybe we have the beginnings of a genetically encoded computer. Okay? Now, when we talk about computers, actually we have to come to Ireland. And we have to talk about this gentleman, Mr. Boole, George, in Cork at the time. And writing this very interesting work, thinking about how human beings tend to understand the universe and how we do that and relate that in ways that are useful to us. And I'm not an expert on Boole. My amateur assessment is he's looking at human language and recognizing that words like and or are incredibly useful to explain what we observe and how we choose to act. So if it's warm and it's sunny, then I might go swimming. Right? That word and is incredibly powerful as a way of coordinating and collating information and realizing something next. Okay. Now I show this also because note that this predates silica-based computers by a lot. All right. So when we talk about computing in synthetic biology, it's not merely recapitulating electronics. We're going way back to something that's more fundamental. The guys who are going to do this are shown here. They're going to take these bits and pieces, they're going to use their labor and change the biology into something new. The thing they're going to make is this element here. It's a piece of DNA that's been engineered to act as a digital switch. Okay? So what this thing does is it blocks the flow of an enzyme called RNA polymerase as it runs along DNA, reading out the DNA. If we flip that object with an enzyme from the bacteriophage that we can repurpose to flip DNA, now that signal can run along the DNA from left to right. I use a different thing from the phage, and I block the signal again. That's it. We call this object a transcriptor because it takes a signal and it controls a current, not an electrical current on a metal wire, but a transcriptional current or a reading out of DNA current along DNA. All right, so it's a three-terminal device that allows for digital switching. How does this relate to computing? Well, if I put two transcriptors in a row, this is the same as Mr. Boole's AND operation. I have to flip the first one, and I have to flip the second one, and that allows the signal to go through. Now, where's the signal coming from? Anything you want. Sense something in the environment, sense something in your gut. Where's the signal going to? Anything you want. Turn purple, float, sink, reproduce, right? It's generic. And it turns out we can implement, by carefully reworking these materials, all of the logic that can be found in Boole's operations. This is a gate called exclusive OR. This would be very useful if you're the French president today. You only do something if one but not the other thing is present, right? Okay. So for the sake of time, I'll push through this. Last year, for example, we implemented all the different logic elements matched to Boole, and we put them inside cells, and they all worked. And they worked via the gizmos of bacteriophage that are repurposed to flip the DNA. Right? Because the enzyme from a phage, if you're a student of phage, it's called an integrase, that's the one we repurposed, and because it's implementing Boolean logic, we had a hacker in the laboratory, and they said, you should call these Boolean integrase logic gates, or Bill Gates, which is what we did. And I'll just show you a quick movie of how these work. So these are cells dividing again, and the middle panel shows the control signal flipping the gate, and the right panel shows the output of the gate. And what's really neat about this logic family is the output is much brighter or much louder than the control signal. So this is the first time in genetic engineering we've had what you would call an amplifier, like a rock concert, where a little signal can be amplified to create a bigger signal. And that's very relevant in computing and control because it allows you to restore information when you connect things up. I know that's a bit esoteric. I'm going to keep going. Okay, so zooming out for the moment, I want to talk about some of the fundamentals underlying this work. Idea number one, biology is not just a science, it's also a material. And we can refine the material. The types of refinements include standardization, akin to what's shown with these rocks here in the aqueduct from Segovia. You alluded to Tom Knight's invention of the BioBrick standard for sharing and coordinating physical assembly of DNA from 2003. That's some of what's happening in synthetic biology. We can also abstract these materials. So note on the previous slides when I was talking about flipping DNA, at no point did I start talking about TAA, TA, CGA, CTC, ACT, ATA, GGG, AGA, or other sequences of DNA. I was talking about flippers. And in fact, we've managed to black box genetic flippers. We've gotten really, really good at that. So I can give you flippers for flipping DNA. And you don't need to know so much about the details of that. That's called abstraction. And it allows people to get access to more powerful functions without having to know all the genetics and biochemistry. Okay, so a little bit of standards to regularize materials supports coordination of labor, and a little bit of abstraction allows us to manage complexity. And now all of a sudden, you might not need to be as much of an expert to do some of this work. And that turns out to be true. So a decade ago, when we started teaching this type of work, we weren't very good at it. But over many iterations, we've gotten incrementally better. Most of the work is still ahead of us, but we've gotten good enough that communities have grown up. For example, the iGEM competition, the International Genetically Engineered Machines competition, this is a genetic engineering Olympics for high school students and college undergraduates, where students can give and get standard biological parts using Tom's standard. We can abstract some of the functions, and sometimes it works. And they make things. It's absolutely incredible. All right, last part of my talk, beauty. The dominant theme when you talk about biotechnology in the United States is the bioeconomy today. And it basically, the story takes this form. Genetic engineering, a generation old, powers actually now 2.5% of our domestic economy in the US. It's split into these sectors: medicines, like recombinant insulin for treating diabetes, foods, GM crops, for example, and stuff. And it's about $300 billion a year of products. It's now about $350 billion a year of products. It's growing 10 to 15% annually. And you can read more about this in Rob Carlson's stuff. If you don't know what stuff is, I'll give you a quick example. You can take the laundry detergent enzymes and adapt them through engineering to work at cold water wash temperatures, and that can save on your domestic hot water heating bill. All right, would be one example. The enzyme subtlysin engineered to do this is shown here. Sometimes it's problematic because it's too powerful and it eats holes in your clothes, but sometimes it works. And when it works and you save on the energy bills, well, in the United States domestically, everybody using this is equal to about 20 of the Gulf of Mexico oil spills in terms of energy savings. Absolutely incredible how integrating an engineered enzyme into our domestic existence can have a meaningful reduction in energy loads. And people are going and going and going. So now we look across all the sectors of our civilization, we figure out how biotechnology can apply to them. We open up one of them, like chemicals and materials, and we imagine we're going to re-implement the entire material supply chain for everything our civilization consumes, moving it from fossil feedstocks into renewable agricultural feedstocks. And this is going to be the future green bioeconomy. Well, I run into two problems with this, challenges, and I need your help with it. The first comes from, for example, some slides I'll show here from John Hoekstra of World Wildlife. When you start to talk about making things with biology, the planet is finite as we understand it. And if I just use the slides here for land use, these are John's slides summarizing how we use land on our planet. 38% now under cultivation, significantly for food, 2% for cities, 5% is desert, 18% looks like this, 14% is parks, and that leaves about a quarter. That's not under cultivation. What do we do with it? Can we do anything with it? It's the source of a lot of our biodiversity and other natural services, quote unquote, to use that metaphor. So the biomanufacturing capacity on the planet isn't now infinite, and I don't see synthetic biology making it infinite anytime soon. Second problem I have comes from my colleague Daisy Ginsburg. She points out that how could a radical technology like synthetic biology result in nothing changing? Here's a slide from a company, Amyris, a nice company using yeast cells to make oils, basically, and you can see that here. And a lot of what's shown here is radical technology applied to maintain existing systems, drop-in replacements, not really changing how things work. And it's the design community, like this science gallery, the exhibition, and the designers working with an iGEM that have begun to open me up to other possibilities. Again, I don't know enough yet to figure out where it goes. But as one example, drawing from this, a team of students engineering E. coli to make pigments. What would you do with this? I wouldn't know. But the designers at the RCA in London could come up with ideas like, let's have probiotic diagnostics that detect disease states in the alimentary canal such that what comes out of you is colored to indicate the specialist you should go see. And that actually seems like a good idea. It seems better than getting a colonoscopy. It seems like it might be affordable to folks who don't have access to first world medicine, but might be able to get access to yogurt. Are we ready for this? Another example, an exhibit here at the gallery are these students who made E. coli smell like bananas or wintergreen. They called it eau de coli. Well, what do I do with this? I don't know. But then I take a project, again shown here at the Science Gallery, where we recognize there's microbes on the skin. And as you can see out in the lobby, you can make cheese from that. And it implies, when you make cheese from human skin microbes, that not only was my mom correct, eat my vegetables because I am what I eat, it also implies that, I eat what I, it's kind of bad. What? How should I think about this? Well, I start taking these things together and I think about the future a little bit differently. I think about a world where this gentleman makes a lot of money representing the distribution of centralized manufacture of petro and plant chemicals in bottles that are then shipped. And I said, no. I want to have centralized, actually kind of distributed design of biosynthetic pathways that produce odorants, right? I can upload the sequence information encoding that to something like iTunes, and I was told this will be called iFumes, okay? And then everybody could download the sequence information for 99 cents a perfume, and you would synthesize the DNA, and you would transform your epidermal ecosystems, and you have distributed manufacturing of living programmable fragrances. You'd reduce the environmental load by quite a lot, right? You'd change the material supply chains. You'd trigger all sorts of reasonable questions about safety, right? And you'd have to address it. What if we think a little bit differently? What do we want? What do we want to put in our DNA? So I'll wrap up by just pushing a little bit and then stop quickly. Double-decker suspension bridges grown in place from the roots of a rubber plant. Fixes carbon while it's grown, passed down from one generation to the next. That seems pretty good to me. How do we do more things like this? Where I live in Menlo Park, I've got a pine tree like this in my front yard. It grows pine cones. I let them fall on the ground and throw them away. That's crazy as an engineer. This is a state-of-the-art nanotechnology that manufactures itself every year from atmospheric carbon and sunlight and whatnot, and I do nothing with this. The city where I live in, 500 pounds per person per year of garden clippings. 32,000 people, 16 million pounds of matter, compostable matter, we do nothing with, except compost it, which isn't bad, but whoa, that's a lot of manufacturing capacity. The world's microprocessor supply is only 1 million pounds of matter a year. Could I re-engineer pine trees to grow microcomputer chips? Don't know how to do it in my lifetime. But I do begin to imagine other possibilities, like taking mushrooms, which we normally think of in California as growing on sawdust and then turning into protein and carbohydrates, and we eat them. All right, that's how it works locally. But the artistic community shows up. Phil Ross, in this case from San Francisco, I can grow other shapes. Mycotexture, he calls it. Well, maybe I could implement program pattern formation within the fungal mycelium to grow the cell phone shapes or the microprocessor shapes. Maybe I could add into that biomineralization, like taken from the sponges in the ocean, and really build new manufacturing processes for making hard materials, things you wouldn't think of as being biological. Okay, so for me, synthetic biology, looking at this at the end, both as a scientist and engineer, includes two types of dreams. For the engineer, I'd like to make living matter engineerable. I don't want to replace the jungle, I want to figure out a better interface with it. There's very provocative installations here, showing what looks to be, for example, a natural landscape that's biomanufacturing space rocket fuel. I'm not sure that's right, but at least it's thinking about that. And then, as I started with the aliens, the default sort of worst case scenario byproduct of synthetic biology is we try things and they don't work, and that forces us to advance our science. What are all those hidden genes doing? I don't know. We got to find out. How do we find out? We try and get rid of them and see what happens. Understanding by building is a very complementary approach. What are the opportunities? Well, we pull off all of constructed biotechnology. I don't even know what all of that is yet. In doing so, we renew our partnership as humanity with nature. And this depends greatly not merely on the industrialization of nature, but on design, ethics, enlightenment, and so on. Thanks very much.
Thank you, Drew. So now we're going to have a discussion. So I invite Hugh Whittall to the stage as well to join Drew and wherever you like to sit. Yeah. So and Hugh Whittall, as Linda said, is from the Nuffield Council on Bioethics. And so Hugh, if you like, I'd just go straight to you and see what do you think from what Drew was just saying. What are the kind of ethical issues that arise for you?
Well, I'm not going to go quite straight there. If I may, I'll just kind of lead in a little bit, and I should perhaps explain, first of all, I'm from the Nuffield Council on Bioethics. Our job is to look at issues coming out of new research in biology and medicine. We bring together, we don't simply say, you know, we're the people who do ethics, we've got an answer. We rather bring together people from philosophy, law, science, medicine, sociology, etc., and work through the factual background, try to develop ethical frameworks relevant to each particular question and context, and then play them through in that context. So there isn't a single framework that we can bring to bear, and we can't just pull one out of a hat.
So you don't have biobricks for ethics?
We have a lot of bricks, but they can be put together in any number of ways, and then they take on different meanings. And I think that in a way kind of reflects what goes on in there. We haven't looked at synthetic biology in particular, but in emerging biotechnologies more broadly. And I think one of the really interesting things that starts to happen, and I think we got a little flavour of this here, is how sometimes it gets talked about in terms of kind of salvation and doom, that, you know, hyping up the potential, but also looking at how it could be catastrophic. And this is because we tend to be driven by visions of the future. What can the future deliver, or what is the risk there? And we sometimes get hard-edged responses about, well, we must ban this or we must regulate it. But I think that, you know, again, one of the things that's become clear from what Drew was saying is that, you know, we're at stages here with these technologies where we are so uncertain. We don't know which pathways are going to emerge. We don't know which things we're going to have to be looking at, whether we're talking about having cheese or coloured crap. You know, we don't know which one is actually going to be delivered some years down the line. We also don't really know what the meaning of those things are because they're quite ambiguous. If we're looking at the potential for delivering medicines, we have a different perspective if we are an academic doing basic research, or we're a pharmaceutical company with the potential for commercial development, or if we're a community that's suffering high rates of malaria. So the meaning of these things will differ depending on those. So the ethical issues that we start to pull out, if we look at a kind of conventional approach, we will start to look at, you know, if we regulate, we want to look at the benefits and the risks. We look at the safety and security issues. But this all depends on kind of measurable risk assessments. And in a way, they just don't work here. You know, they're not knowable. And if we try to drive through a kind of classic precautionary principle, which is, you know, taken in its kind of extreme form, don't do it until you know it's safe. Well, we just stop. So I think this, in a way, doesn't work. And so what I'd like to do here, and in a way it's fantastic to be here because this is exactly it, is actually promote the idea of what we've referred to as a public discourse ethics. So this is the idea that the ethics that applies falls out of a public discourse rather than it being something that's ready-made for us. So, you know, we have an interest in developing, in identifying, you know, where the public interest is, what the public good is that we're looking for. We can see that in many circumstances, the development of science is captured by elites, whether it's a scientific elite, political elites, or those that are driving this economics agenda that says what we want to go for is that which gives us economic growth. So we need public discourse to kind of get away from those capturing by elites so that we can know what the social concerns are that we want to deal with. We can discover the ethical values that we want to work with, and we have got lists, we have got our ethical bricks, and we can talk about solidarity and sustainability and equity and justice, which are the ones that we've talked about. The US Presidential Commission, in looking at synthetic biology, talked about beneficence and responsibility, which is about behaviours, and about democratic deliberation, which again is about a public environment in which we discover these things. So really this discourse ethics is about a constant review, a discourse that is in a public space of reflection and return, so that if we're going to use regulatory mechanisms, they've just got to be reflective and responsive and vigilant to the risks that might fall. But really, this is not yet the time to try and nail it down.
Mm-hmm. So you mentioned there the precautionary principle, which is basically the don't do it until you're sure idea. But so what I gather from what Drew has been saying as well is that this is it's already happening and it's already possible. So is it more the case that this is there and it's a matter of how we deal with it and react to it? There is the option of not doing it all doesn't really seem to be there.
I think that's right, but I think that there's one approach to the precautionary principle, which is this: don't do it until we know it's safe. But if that were the case, we would never have had internal combustion engine, any number of things that we can think of. So I think that I think that we should be more cautious about how we use precaution. This is work that's already happening, and I think it's to the great credit of Drew and other colleagues of his, and of people who are certainly working in the UK. They've put themselves into a public space and said, let's talk about this now, at an early stage. Mm-hmm. And I think that we've got to embrace that and take that opportunity, because it's not run away from us. And the things that are going to come out of this, we've yet to see them, but let's start thinking about it.
Yeah. And so, Drew, you made one comment, you don't need to be an expert to do this. And this is kind of one of the driving philosophies, it seems, to simplify and make it as abstract as possible and have the blocks. And I know there's at least one guy in the audience here who has a lab in his garage. There he is over there. And so, so the idea that, this is a technology that in a sense has been released, and it's there, and we can't. And so, would you agree with that idea that this is already something that's completely out there and could be done by anybody potentially?
Yes. With qualifiers, because one of the metaphors that's drawn, a historical metaphor, is as was happening in synthetic biology is akin to what happened with the homebrew computing club in Silicon Valley in the 1970s. And that people went into their garages with the tools of the day in electronics, and out came Apple, right, and other things. And that's true. In biotechnology, we don't have yet the same investment or maturation of the infrastructure or tool base. 1970s electronics is 30 years of investment in electronics, silicon wafer manufacturing, the transistor logic data book from Texas Instruments and others. And those things worked incredibly well by the times people showed up to make the personal computer, to take the computer to themselves. So it's a challenge, I think, for a lot of the extra-institutional, independent biotechnologists to have the same rate of progress, because things like the BioBricks program, things like working on abstraction, are frontline research questions that haven't been worked on for decades. They're almost brand new, and if I were to be honest with you, it's not until last year that I could report and feel confident that we figured out how to make really good standard biological parts, meaning when you put them together, they'll behave as expected. And we just didn't know how to do that in 2008 or 2005. So it's certainly the case that the capital costs of setting up a laboratory are affordable, very, very different, and nice for biotech relative to other types of technology. But it's early in many respects with respect to the maturation of the technology. So I wouldn't want to over-represent, that somebody's going to go make a 8-bit computer or synthesize a hemorrhagic fever, right, or something like that. I don't see that as being trivial yet.
So you don't see, because that would be a fear, that could be a fear of bioterrorism, that somebody's going to create, using this type of technology, create something extremely dangerous. So you don't see that as a realistic fear right now?
I have a variety of ways of looking at that, ranging from to represent it will never happen, and that's more of a political approach than a technical assessment. Leading by declaring something to be just so bad it cannot ever happen. And that sets a frame of action.
So morally bad, you mean?
Yes.
Okay.
The flip side of it is, in San Francisco, so far this year, we've had 16 deaths from H1N1, the variant of flu, which comes from nature. Shows up every year. It kills 30 to 50,000 people in the United States. that has nothing to do with my actions or the iGEM students' actions. If anything, it has to do with our inaction, right? And within the research community, the biomedical research community in the United States, there's a huge fracture right now between whether we should be exploring things like diseases so that we can better defend ourselves against them versus we should just wait for something to show up and then react. And people are very discomforted by the idea of taking risks to find therapies.
Yeah.
I think that, I mean, that all kind of points back in a way in the same direction, which is to say, well, I mean, these are legitimate questions and concerns in a way that there may be some risk of this or this sort, although we haven't seen, you know, natural pathogens used in ways of terrorist activities. Why would they go to the trouble of kind of creating new ones? But so the important thing in a way is to say, well, let's keep this in a public space so we can watch. And if there is a public interest, and there is, whether it's about trying to get the benefits or guarding against the risks, then we need to do, it seems to me, we need to do two things. Is one, be involved in the research from a public perspective, and to be involved in a wide range of research because we don't know which ones are going to progress, so that we can introduce the social interests that we have into capturing the ones that are going to deliver the benefit, but we're also in a position to identify the points where the risks might arise. So it's a kind of dynamic process of working with this, but doing it in public spaces.
Yeah.
When I say public spaces, I don't mean, you know, out in the middle of the park. I just mean in places where the conversations and the discourses are not captured by people with particular partial interests, but there is multiple voices involved in that discussion.
I think what I would agree with what you're saying as being a best approach. I would just simply acknowledge that it comes with responsibilities. You know, when you do things in a public space, when you build open technology platforms, you have to presume that people will then actually go extend what you're doing and take it in ways that are new and different from what you would ever imagine. The most recent example of this for us was the Kickstarter-funded project, crowdsourced funding to make a glowing plant. Right, so a team of folks repurposing lab equipment into a mobile container platform raised $500,000 to make glowing plants from a crowdsourced funding model. Nobody was ready for this. Right? But we should have anticipated it. And we really weren't prepared. And so it created various types of blowback. For example, the funding mechanism, which could be a very interesting funding mechanism for taking biotechnology forward in a democratic sort of way, a distributed way, in terms of raising capital, so you don't have to be a capital elite to fund a project. Not being ready for that caused the host organization around fundraising, Kickstarter, to disallow future projects involving genetic engineering. Right? So, you know, I agree with what you're saying as the approach, but sometimes it's hard to keep up with it. Sure.
So this actually leads on to one of the questions that we'd like to ask the audience with your clickers again. So it's question 2 on the list up there on the computer, please. So, and this is, so who should design life? So this question, you know, so how open should this be? And so we've given a few suggestions there. Notably, we don't have the option 5, which is nobody, keep your hands off. So, or me. So we have scientists, designers, engineers, or some kind of interdisciplinary group. So if you could vote on that, we'll just get a feeling for what the audience think of this. And so you can pick one of those, and then we'll have a bit of a, I don't know what you guys, Hugh and Drew.
Honestly, well, is everybody done voting? I don't want to corrupt the experiment.
I don't care for the question very much.
Yeah.
You don't.
But if you had to answer the question, what would you pick?
It would come down to D, I guess, if I had to answer between them.
I would probably pick B.
Designers.
Yeah, because
like Daisy Ginsburg, so the curation here that you mentioned.
I mean, I think they represent the professionals who are best at assessing what people might want.
What do the audience think? Let's have a look at this. Okay, there we go. Interdisciplinary.
Not the engineer. Not the engineer. There's one engineer here.
We have 100 clickers, so we can count the number of people who voted for engineers very easily.
Name and shame.
So there's fingerprinting going on, so we'll know who you are later. But so you don't like this question?
Well, I actually would rather we didn't use the idea of designing life, creating life, because I think that there's something happening here which is in many ways a creative activity. But I think that we need to kind of go back and have a kind of big conversation about where we're putting the borders of life and not life. And I don't think that's very helpful to do it quite like this at this point.
Okay. And I'm going to, if there are any questions in the audience, we'll look to you guys for questions now. But so when you say design, it rhymes to me, because I work in evolution, this whole idea of evolution versus design. And I know, Drew, you've made a few comments on the tyranny of evolution, I think you called it.
Sure. Well, I mean, there's Tom Hardy's poem, right? I am the family face, flesh perishes, I live on, carrying trait and trace through time to times and on, and leaping from place to place over oblivion, representing, how your offspring might look like your ancestors or you. And that is associated with this mechanism of direct descent from one generation to the next, and mutation and selection and evolution. And it's such a constraint. It really is a hard constraint on the architecture of natural biological systems. So the playful turn of phrase is basically evolution is a tyrant. It gives us mutation without representation. And in Boston, where this phrase came up, the idea was to take bottles of phosphoramidites, the chemicals you use to synthesize DNA, and dump them in the harbor and have a party, to liberate ourselves.
DNA party.
Yeah, that's right. Liberate ourselves from the tyranny of evolution. Because with synthesis, you can make a piece of DNA and use it, and if you want to make another copy of that piece of DNA, you can synthesize it again. It's not constrained to direct descent. Do you see what I mean? And it's a provocative statement, perhaps, and maybe overly so.
You can also design the DNA. You can get optimal DNA sequences through evolution.
Totally.
So some designers use evolutionary algorithms to design, better nose cone of a plane and different things like this. So
and better proteins.
Yeah.
Right. So absolutely, evolution and rational design are totally complementary. But could I, Hugh, return to you? I don't have a problem with the word design. I do have a problem with the word creation as an engineer. Creation implies this godlike ability to know everything, to be able to absolutely control matter, to have an unlimited budget, right? As an engineer, I don't have any of those things. I have a budget, I have an imperfect understanding of the universe, I have a very crude capacity to change stuff. So I'm very comfortable with construction as a verb. So I wonder if that's at all.
I think again, we're using these things in different ways. I'm talking about creative in the sense of an artistic creativity, which is not to create something de novo, but to use things in a way that has got a creative element to it. So that I think is where we kind of need to be careful about this. We also people come from different cultures, and I don't mean geographically or ethnically, but when a few years ago I was in a nice meeting with the Synbio community in the UK, and what was emerging was that the biologists were going, well, it's great that we're all working together, but I'm not sure about these guys. They don't really understand, regulation and risk. We work with dangerous pathogens in the lab all the time. We know about these things, but those engineers, they don't really. And the engineers are saying, don't know about those biologists. They don't really understand about risk. We build bridges. We know about this. And so there are kind of these cultures that come together. And what we're doing now is putting them in this place where we're saying, well, because these have come together, we now are creating, we're designing, we're doing life. And I think that we just haven't really worked out what the territory is, what the language is, what the concepts are, what the boundaries are. And this is where I think the artists are doing some fantastic work, which is working at boundaries, which I think we had a conversation about this a little earlier, and trying to understand those boundaries. And so I don't think they've yet been settled, which is why this is fantastic what's been going on here in the exhibition.
That's great. Do we have any questions? If anyone has a question in the audience, raise their hands. We have a question here. There's a microphone just coming. Just wait for the microphone. Thank you.
It relates to the question, who should design life? It actually is the opposite in a way. Who should control life? Who should control life? And so I wanted to ask your opinion on certain kinds of control, like governmental control, international control, control by academics? What do you think about who should regulate?
If that comes into, does that come into synthetic biology for you at all? In terms of, you're talking about controlling human life in this case.
Yeah, I guess it maps practically, perhaps, into process and product regulation. So, for example, should you regulate DNA synthesizers, the tools that take in information and sputter out DNA molecules? Alternatively, should you regulate products? Should you regulate a glowing plant as a type of artifact produced via a combination of tools? And depending on who you are and where you're sitting, sometimes the answer is yes and yes. For example, DNA synthesis technology, at least in the United States, is primarily regulated at the federal level with an eye towards biosecurity as the driving imagined risk factor, right? Whereas engineered organisms that might be released, if they're regulated, to the extent they're regulated, are regulated from the perspective of human or environmental safety as a product, not as a process. So I think you're posing a very good question, and the answer is kind of as complicated, depending on what aspect of the workflow you're looking at or what types of artifacts are being made.
I just entirely agree with that, and just to say on top, I think that it would be at this stage, and probably and possibly indefinitely, a mistake to imagine that we can simply design a single regulatory structure that will do it. I think that it's going to have to be a kind of more organic, responsive way of managing it, because we're looking at across national, international boundaries, as well as disciplines, and as well as types of activity, types of product. So I think that we need to really kind of alert to how we can have a reflexive type of regulatory response.
I would agree with that. Ken Oye's group, by the way, at MIT, has written what's called a concise guide to regulation in synthetic biology. Last name is Oye, and you can find that on the internet. It's 32 pages, summarizing existing regulation of various aspects and products of synthetic biology. And I mention that not to argue that it's sufficient or comprehensive. It's not. But it also is a existence proof that we're not operating in a vacuum with respect to regulation, just the opposite. Okay. And by the way, many people would argue those regulations are killing people by hindering action.
By stifling science.
Yeah. I think that's part of the problem with the precautionary principle as applied in a strict form, because one of the factors that you must take into account is that if you don't do that, then the status quo is not a neutral
the cost of doing nothing
position. Yeah.
Okay. That's an interesting point as well. So we have another question here from Cahill.
So, not meaning to rehash any of everything that's been discussed so far, but how different do you think the discussion we've had to this point would be if it weren't for the fact that we tend to fall into the metaphor of product? Like, we don't view a pet as a product, even if it is a thing that is created by someone else's biological product, if you want to look at them that way, but we don't see them as products, and living things are seldom viewed in and of themselves as products. So how do we view regulation if you're not talking about production and product, but rather just a living thing, if it's just a thing unto itself?
I probably won't be able to answer your question very well, but I think that one thing that it's worth bearing in mind is that the word regulation is used ever so easily. I would rather, in a sense, step away from that and just talk about broader ideas of governance that we can work in many different ways. And you can have self-regulation of the individual simply through the moral activity. You can have professional codes which are self-regulatory. You can then move up to a more kind of codified set of instruments of regulation into legal instruments and prohibitions. And so I think that you can have a kind of matrix of types of activity, types of product, types of and then types of regulation, and the ways regulatory mechanisms and the way that they can be enforced. Now, this is, again, just goes back to complexity, and I think that we have to be ready to deal with complexity and ambiguity in a way that it feels uncomfortable for policymakers.
Let me amplify that with an example, but just to your question, I think we've got products all over the place and they're recognized. Glowfish, various crops with particular traits. It does get confusing from a property rights perspective when you start to think about who owns those things. Is there patent exhaustion as a particular example? And the Supreme Court in the US at least is working through all of this. But we do have products, at least in my limited position. In terms of governance, just as an example, I showed that figure of those folks in iGEM. The original proposal for that student activity wasn't called iGEM, it was called Bug Wars. Teams of students would engineer microbes to kill other engineered microbes. It would trigger this adolescent excitement for combat. It'd be very easy to judge. We wouldn't have all these judging difficulties because you just put everything in a tank and let them duke it out, and you'd go out for a pint and come back and see who's left. They win. But in the context of governance, when you think about all our options for shaping culture and community, right, it's kind of a good idea that we didn't maybe go with that we went with the International Genetically Engineered Machines competition. Not that that's perfect, but at least it's creating something existing in a space. We call that event the Jamboree, thanks to the Scouts, right?
Was there not some discussion as to whether you would use the term synthetic biology or some other thing?
All the time, the time. And it still happens. In the US, we're going through a relabeling of the field right now at a political level. But the thing that shut down the renaming conversations was the following idea. Let's rename the field Shiny Happy Biology. And then we won't have anything to worry about. It'll just be Shiny Happy Biology. And that ended those conversations because it was apparent to researchers that there are things to talk about, and you shouldn't create labels that preemptively push things under the rug.
But this is sorry if I may. Again, there was some interesting conversations that I recall about whether we should talk about synthetic biology as being kind of just incremental developments on the stuff that we've done before, which is what we might do if we're talking to the media and to the public. Or whether we should talk about it as a complete new paradigm shift, a whole new paradigm over how we do science, which is what we should talk about if we're talking to funding agencies. This does become a kind of tactical and political thing where, the words can make a difference, and you don't want to use the wrong language in the wrong environment. In a way, it's wrong that we should do that, and again, it needs to be exposed to the public light if we are going to have actually open, honest conversations and build the trust that's going to be needed to keep moving.
And I think there's this idea of governance and of self-governance actually links in with another question I'd like to ask the audience. So it's, do you trust scientists? So it's question 3 here. Do you trust scientists to make ethical decisions about synthetic biology? And I'm going to ask you other questions about other people you might trust. So this is just about scientists. So it's basically, do you think science can be self-regulating, and which is it's a principle that some people do adhere to, the idea that the scientists know what the risks and the potentials are, therefore they're well placed, but maybe they're also over-enthusiastic and they
like to ride motorcycles.
And they like to ride motorcycles and graze their knees as they take the corners with such speed. So. Once we get, we're nearly ready. I don't want to preempt your voting too much. Yes, so let's see okay, now we've got you. Let's see the response. There we go. More people don't trust the scientists. Are you surprised by this, Hugh?
Bearing in mind the answer to the earlier question, where people kind of really wanted to see more people engaged, an interdisciplinary approach. It maybe reflects that a little bit. So we'd already seeded the idea in people's minds.
What would you think on this particular question, Drew? Do you think it should be self-regulating?
I think some things have to be. Not everything can be, but some things have to be. And it's again, when you think about, I think governance is the right word. It's a suite of approaches. Some types of issues, like if you take safety as an issue, we're changing things with biology. How can we be as safe as possible? If you don't have the people actually doing the work, taking on some type of responsibility in initiating the work, you're going to have a hard time being safe. At the same time, if you don't have some type of oversight mechanism that accounts for, are people actually having conversations about what they're proposing to do? Are they making sure there's appropriate safety protocols and procedures in place? Then you'll get a lot of dangerous leaks through the self-governance approach by itself. So it's a combination of approaches in practice that seems to work. I'll leave it at that.
Okay.
Yeah.
Are there any? There's another. So there's a question. The audience at the back there. Yes. Yes.
Hi guys. This is coming back to ethics. As someone who, about a decade and a half ago, went to court and challenged the precautionary principle in relation to food safety and cheese, but now I have the unenviable position of being a Green Party spokesperson for agriculture. So I am coming in conflict with my dear friends in science and my dear friends who have great concerns maybe for other issues. So this is not just a question for the panel, but a question for everybody here, is how do we address this huge polarization that exists now between people who have environmental concerns and the polarization between them and the scientific community? And I really like the idea of the difference between responsibility of corporations and respect for social justice. That's a fascinating question. And secondly, what does the panel? How does the panel think we can address this? Because I get it huge on social media. I get an awful lot of abuse for being green. Yet I am, as most people here would know, fascinated by science and GM. But I am at the risk of getting expelled from the Green Party. That's how do I not get expelled from the Green Party? Is my question for the panel.
So isn't there an interesting history with the Green Movement, though, that the Green Movement used to be more scientific? We had an exhibition here a couple of years ago, Green Machines, and it was the idea of taking a scientific approach to green attitudes. So Eamon Gormley launched it, so maybe you can remind him of that.
Is a genetic alteration of John Gormley.
Yeah. The green guy. What was his name again? Anyway, John Gormley. Eamon Ryan. That was it. It was Eamon Ryan, not John Gormley or Eamon Gormley. Anyway, but so there is, I think, at some point in the Green Movement, there was a greater appreciation for science as a way to achieve some of the green goals, and at some point that has been rejected.
That's part of the Green Movement. There just seems to be a huge battle between environmentalists and science, which has been polarized by some people in the scientific community and some environmentalists.
I mean, I reckon, I could probably talk for a long time about this, and we've been around a couple of questions where we've seen these things. We did a report some years ago on the use of animals in scientific research, where you got polarization of views, and you've got to get onto territory where people can talk together. I mean, that's kind of easily said, but it's becomes about trying to create a culture of openness and honesty, that scientists are more honest and open about the limitations of what science can do, because sometimes it's simply talked up in kind of overhyped terms, and I think we have to, we will get more trust, if you like, if there is a little more humility sometimes. And on the other side, I think people also have to be willing to listen. All we can do is keep encouraging this kind of public space in which these things can take place. And policymakers must keep all of those people in the conversation. So that's a kind of, maybe that's a kind of optimistic hope in the face of known reality.
Well, we have another question, actually, so similar to this one, but do you trust policymakers? Instead of, you've mentioned policymakers, so we're 57% do not trust scientists, but what about policymakers? How would you feel about policymakers making ethical decisions about synthetic biology? And we'll see what you guys think before we destroy the validity of the vote by talking about it too much. We're nearly there. Everybody nearly got a few more votes to come in, I think. Okay, let's see what we got. So scientists are better than policymakers. That's one thing. So it's that's a faint compliment, isn't it?
Yeah, we were called to testify before Energy and Commerce in the U.S. House of Representatives on synthetic biology. And it was right at the time that the BP oil spill was taking place, and the CEO of BP was also testifying. And one of the questions we got from the ranking representative from Texas, which was a good question, but it sort of illustrates in a way, could you engineer a genome that would predispose folks to vote Republican? It's quite an astute question, because the answer is yes. It triggers all sorts of ethical questions. But I think to the earlier question, we face this puzzle where, political systems struggle to deliver needs and services, satisfy, and the scientific community, the research community, is oftentimes incapable of carefully articulating capacities and where we are. And when those two things meet, the politicians, quote unquote, are happy to throw something over the fence, and the starving research community that needs funding is very happy to say, yes, I'll solve your problem, right? And the reality is somewhere in between. And I'd love to figure out ways of, for example, imagining Greenpeace or the Green Party or civil society organizations representing very diverse views. Not all of them, because some of them are really diverse and are going to be extreme, but not all. Figuring out ways of exploring partnerings and partnership that's not just packaging, but actually working together on something. One of the things that's oftentimes true about technology is if you don't own it, you don't have leverage. Now, owning could be a private type of ownership, or it could be a commonwealth ownership. And so I think exploring ways of working together, this isn't going to be effective if you're totally against the morality of changing the DNA that exists on Earth. That sort of disallows all sorts of types of, but maybe there's other types of partners. I don't know.
Okay. So we have another question. There's a lady here, their hand raised. There's a microphone coming. Yes.
Just in relation to what you're saying, do you think the bioengineer is adequately trained or funded to determine the implications of the potential impacts of their new combinations of living organisms on the natural and human environment? Do you think the bioengineer needs to be trained in a different way to deal with what he's working on?
I think there are types of potentials which are different in biology compared to other technologies. This capacity to reproduce, this capacity to evolve, right? This capacity to freak everybody out if you do anything at all and make any progress whatsoever. And, I think that both the training and the resourcing is woefully insufficient. From a technical perspective, I could give you an example of a project that I think would be interesting to explore. The natural genetic code that creates the mapping between DNA sequences and proteins is itself a product of evolution, which means it's been evolved to support evolving. Wow. Now, I'm working within this framework, within this tent of evolution. Could I design a synthetic genetic code that is what an engineer might call fail fast? When the rocket launches and something is not nominal, you blow up the rocket before it goes and crashes somewhere you don't want it to crash. So this would be called a fail-fast system. Could I make a fail-fast genetic code where every point mutation is deleterious? Turns out you can imagine it. You have to go from 3 bases to 4 bases, 256 codons. You have enough empty codons so every point mutation would map to a null. It gets a little techno mumbo-jumbo, but holy cow. Now, by the way, these organisms would probably barely grow at all. But all of a sudden you can see, wow, there are ways of thinking about what we could realize as best available technologies from a safety perspective, and we haven't even begun to have conversations about exploring them or developing them, let alone funding them or helping other people to think about it. So I think your question is a brilliant question, and again, my response is not close to being adequate.
Okay. I want to say one thing quite plainly. I don't think scientists are unethical people. I think that they are, just like the rest of us, make good ethical decisions all the time. We all do that all the time. We don't always know what we're doing when we do it. We don't always deconstruct it and understand how it works. And I think we could all benefit from having a little more training and understanding about how we do make ethical decisions, whatever context we make them in. And certainly people working in the life sciences, I think that there's probably a lot of room for enabling them to have some degree of reflection on those things, but also to work alongside other people who are bringing in just other ways of looking at the things that they're doing, and whether it is social scientists or people working in humanities or whatever else. I just think there's a lot of benefit to be had from all of us opening ourselves up to other people kind of looking in and talking about it.
And it's a start here tonight as well. I think there's another question here near the front, this gentleman here.
Thanks. I just think there's one large elephant in the room that I think you alluded to slightly, Drew, when you talked about if you don't control your own technology, you don't have any leverage. And that's around money and corporate interests, which very often trump other sorts of interests when these new technologies are emerging. And I'd particularly just like to get your view in terms of how open source kind of ideas or concepts are sort of emerging. How much leverage or strength do they have in the area of synthetic biology at this point? I know that, say, Richard Jefferson in Australia was doing some work on this a long time ago, but I'm not aware that it's gotten any much momentum or kind of purchase.
So, a lot to say on that. Let me just try and create some bounds for the sake of time. Free-to-use technologies can enable people as new actors to come in and make their own leverage, but they can be just as readily exploited, so to speak, by well-resourced capital. And there are fine examples of that, using free software, leading into web services and organizations, like Google and Facebook. Think of them what you will. At the same time that the free software and open source software movements allow many people to do new things, creative things with software and compete, perhaps. But note that these networks are shaped by network effects, ways of structuring transactions and relationships. And I'll turn to biology in a second, but I want to mention one of the things that I'm spending a lot of my time thinking about from sort of cultural architecture perspective related to your question. It's the so-called Joy's Law. Give credit to Bill Joy. No matter who you are, most of the smartest people will work for somebody else. And the reason I find Joy's Law to be so interesting is because I think it applies. He was at Sun Microsystems when he stated this, apparently. And the hidden question in Joy's Law is, so what do you do about it? And at that time at Sun, there was a balkanization, a lock-in between hardware and operating systems. So if you were a software developer, you were developing for one platform and not the other. And the hack, the what do you do about it at Sun, was you implement Java. The Java programming framework that basically allowed people to write software in Java that could run on any of the major platforms. And that allowed indirectly every software developer to choose to work for Bill, right? Even though they didn't work at Sun. So Joy's Law, I think, applies to the future of biology and biotechnology more than any other science or technology. Most of the smartest biologists will work for somebody else. Most of the best biobricks engineers will work for somebody else. Most of the needs and opportunities that could be addressed with biotechnology will exist somewhere else. Most of the problems I would cause would hurt somebody else. It's just all these network effects all over the place. Now, Rich Jefferson has done some heroic work with the BIOS program. He tried to use patents as a legal mechanism to create an open source platform. The problem with patents on a very straightforward basis is their transaction costs are way too high. You just can't afford to participate. Through the BioBricks Foundation, we've taken the other approach. We're using the public domain and basically promises to not assert property rights to create a free-to-use commons of genetically encoded functions. And we've just started doing that. It's not very popular. I'm like the major contributor of this. We've had one company contribute free-to-use fluorescent proteins, and not many people want to give things away. So it's a real weak spot, I would say. We have a lot of work to do. And even if you were to take it forward, I think these network effects are going to be capable of being captured if we're not careful in ways that might not serve everybody.
I think we have one more question here, and then we'll be wrapping up soon. I'm going to ask you to clicker for one last question as well. It'll come on, yeah.
Yeah. Some of my colleagues have been working on a project called Reflexive Systems Biology for a few years, where they looked at not only systems biology, but also synthetic biology and some other more traditional forms. And one of the things they found was that there's very little interaction between different forms of biology, and sometimes people can be working on the same problem from different angles, but not actually discussing it very much in between them. And it struck me that most of your references tonight have been to computers and engineering and not so much to biology. So I was wondering, when it comes to languages and these things, if that could have some kind of,
It's a good question. I mean, part of what's happened is you have to create space, and a cultural space, in order to allow for new ideas to be explored, even if they might be redundant with other approaches to the same puzzles. And a lot of what's happened in synthetic biology is to just create a little elbow room, to reconfigure a little bit. As soon as that happens, there's another quote, I can't remember, the most radical revolutionary becomes a conservative the day after the revolution. As soon as you do something new, you want to lock it down. There's a strong tendency to continue to just control what's already happened. And so, as an idealistic sort of response to your question is sort of, how do you sustain, pushing to create room to do new things, yet remixing with the old? And it's not trivial, it's not easy, it's not well done. And to be honest, a lot of researchers are in a hand-to-mouth survival mode in terms of funding and this, that, and the other thing. So it's very, very hard to afford to open up interfaces when you're just trying to protect, turf.
And I think that's true in many, many disciplines. I mean, I certainly see it in the humanities areas, where people end up having to kind of specialize and super specialize, and then they can ill afford to share too much because they have to have something unique to sell, both to funders and to publish. And so there's a kind of drive that is in the environment, is part of the culture that I think we need to work against, is a research culture that actually is driving people in the wrong directions for all best intentions. The funding, the professional progress, it's all actually working against the interests of the kind of broader interests of science.
Yeah, that's very. So I think we've kind of come to the end now of our time. I want to get you guys to do one more question, which is just to ask you once more whether you think synthetic biology is a good or a bad thing. It was question 10, if you can pull that up. And hopefully you know what it is by now. But also I was wondering if Hugh and Drew, if you would like to just, if you have a final comment you'd like to say just before we finish, any message you'd like people to take home with them, or anything to wrap up with.
My message, I think, has been a simple one, and I would just like to repeat it, which is that the thing that is going to be most important here is the public space, and people in a public space where many voices can be heard in considering, what should happen, what should be funded, whether it should progress, and I think that we all kind of have to have some responsibility of trying to understand where that public space is and engaging with it. And so, it's not just today, guys. When you get out there as well.
Okay. Well, thank you. And Drew, do you have anything?
Well, I guess I'd just offer an observation and a thanks. For the rest of the century, our lives, we're going to learn more about biology. The scientific community is going to bring us those insights. We're also going to get incrementally better at engineering biology over and over and over again. So whether you like it or not, it's hard to imagine stopping that broadly. And the question then becomes, since biology is so central to our very being, to nature, to our civilization, what do we want? And I thank the Science Gallery for creating the venue to explore this. I hope and would ask that each of you, as you go out with your families and networks, you help other people to engage with the stuff of biology and to think about these questions. Because I think the type of dialogue and engagement is critical to ensure that we don't merely default to some future, but we create the future around this, which is beautiful.
Thank you. That's a very interesting idea. So this remains for us to see what you think of synthetic biology, Now, yoo-hoo! We have fewer don't knows, which is already a very good outcome. And so that'll be thanks to Drew and Hugh. So if you could all join me in thanking them now. Thank you.